When you think about your brain, it's just this constant storm of electrical signals, right? They're telling your body to move, to think, to feel, everything. But have you ever stopped to wonder what tells those signals when to, well, stop? That's where the brain's brakes come in. Today, we're diving into the powerful off switches that keep our minds from just spinning out of control. They're what allow for everything from a focused thought to just holding your hand steady. So what you're looking at here is a synapse. It's that tiny, tiny gap where neurons communicate with each other. And it is a place of nonstop electrical and chemical chatter. But all that activity would be totally meaningless, maybe even dangerous, without a way to quiet things down. I mean, uncontrolled firing, that's just chaos. So how does the brain actually pump the brakes? The answer lies with a special class of molecules, the inhibitory neurotransmitters. So here's our game plan. First, we're going to get a handle on the basic idea of the brain's on-off switches. Then, we'll meet the star of the show, a little chemical called GABA, and see how it's actually made. After that, we'll look at the super intricate locks that GABA fits into and introduce its important sidekick, glycine. And finally, we'll explore what happens when we start tampering with this very delicate system.
All right, first up, the on-off switches. The whole principle here really comes down to one word: balance. For every single action in the brain, there has to be a reaction. For every signal that screams go, there absolutely has to be one that says stop.
Honestly, the easiest way to think about it is like driving a car. On one side, you've got the accelerator. These are your excitatory signals. And the main one is a neurotransmitter called glutamate. It's what tells neurons to fire. But on the other side, you have the brake pedal. These are the inhibitory signals, led by GABA, telling them to stop. It's this constant, beautiful dance between the gas and the brakes that allows our brains to do everything they do. And you gotta understand, GABA isn't some minor player here. Not at all. It is the most common inhibitory neurotransmitter we have. A full a third of all the connections in your brain rely on GABA to pump the brakes. That is a staggering amount of control that's dedicated just to keeping the entire system stable and in check.
Okay, so if GABA is the brain's main brake fluid, where does it even come from? Well, the answer is surprisingly and kind of beautifully elegant.
Now, this is what's so fascinating to me. The brain builds its main stop signal directly from its main go signal. It's incredible. GABA actually starts its life as glutamate, the brain's accelerator pedal. Then, with the help of one special enzyme, a single chemical step just flips the switch, turning that go signal into the stop signal.
That crucial enzyme is called glutamic acid decarboxylase, or just GAD for short. Think of GAD as the molecular mechanic that pulls off this amazing conversion. But for GAD to do its job, it needs a very specific tool. It needs a cofactor that our bodies make from vitamin B6. And that one little detail, as it turns out, has some serious life or death consequences. This quote here, it highlights a really tragic, real-world event that proved just how critical GABA is. Back in the 1950s, a batch of infant formula was accidentally made without any vitamin B6. And without it, the baby's brains just couldn't produce enough GABA. The brake pedal was basically gone, and that led to a total loss of synaptic inhibition. The result was just uncontrolled brain activity, which caused these devastating and sometimes fatal seizures. It is just a stark, powerful reminder of how important this system is.
Okay, so making the GABA molecule, the key, that's only half the story. Because for that key to work, it needs a specific lock to fit into on the surface of the next neuron. And we call these locks receptors.
And GABA has two main types of locks it can fit into. First, you've got the GABAA receptors. These are kind of like a simple deadbolt. They're direct, they're fast. When the GABA key turns, a gate just swings right open. Simple. But then you have the GABAB receptors. These are more like a complex electronic lock. The GABA key starts the slower, more indirect process. It kicks off a whole chain reaction inside the cell that eventually leads to the same result.
And just look at them. You can see their structures are completely different. On the left, the GABA receptor is this big, complex thing made of 5 separate protein parts. But on the right, the GABAB receptor is just a simpler pair with 2 units working together. And these totally different structures lead to very different ways of putting on the brakes. Let's zero in on that GABA receptor for a second. See how those 5 parts form a little channel or a pore right down the middle? Well, when GABA binds on the outside, this channel opens up and allows negatively charged chloride ions to just rush into the neuron.
And this flood of negative charge makes it much, much harder for that neuron to fire. That's inhibition in action. It's direct, it's simple, and it is very, very fast.
Now, this is where it gets really important for us in the real world. This single receptor has multiple places for things to dock, not just for GABA, but for all sorts of other molecules. I mean, think about it. Common drugs like benzodiazepines, like Valium, anesthetics, and even alcohol all have their own little specific parking spots right here. They don't block GABA, they just make its natural effect even stronger. They're essentially pressing down harder on the brain's own brake pedal. Now, the GABA receptor, it works in a completely different way. It's often described as a venous flytrap. So when the GABA molecule gets caught in the trap, it triggers this change in shape. This kicks off a whole cascade inside the cell, activating other proteins that ultimately put on the brakes. The end result is the same, inhibition. It just takes some more scenic route to get there.
So while GABA is definitely the big star of inhibition in the brain, it is not the only player on the team. There's another, smaller but equally vital inhibitory neurotransmitter called glycine. You see, glycine is a specialist. While GABA is handling the complex stuff up in the brain, glycine is the undisputed king of inhibition down in the spinal cord and the brainstem. It's actually responsible for about half of all the braking signals in these areas that control our most vital functions. We're talking everything from basic reflexes all the way up to breathing itself.
And glycine works in a way that should sound pretty familiar by now. It binds to a receptor, that opens a channel, negative chloride ions rush in, and that inhibits the neuron. But here's the really dangerous part. The infamous poison strychnine is a perfect fit for that exact same spot. So it gets in, it blocks the channel, and it stops glycine from doing its job. The brakes are completely cut. And that leads to this catastrophic loss of inhibition, causing violent convulsions and muscle spasms.
So all of this brings us to our final section. What happens when we intentionally start messing with this incredibly delicate balance of braking and acceleration by using various substances?
Yeah, this list should probably look familiar. This is basically a who's who of substances known for their calming or sedative or sleep-inducing effects. And they all feel different, sure, but they all produce these similar outcomes for one simple reason. They target the exact same fundamental system. Every single one of these works by binding to that GABA receptor and just amplifying the effect of the brain's natural brake, GABA. Let's just focus on the most common one, alcohol. Why does it make you slur your speech or mess up your coordination or make you drowsy? Well, at a molecular level, it's because that ethanol is binding to your GABAA receptors. It's artificially slamming on the brakes all across your brain. And that leaves us with one last, really crucial question to think about. This delicate balance between go and stop is what creates the entire symphony of our consciousness. And yet, as we've seen, the stop side of that equation is incredibly easy to influence. A simple pill, a common drink, it can alter this fundamental process. So what does it really mean that we have such direct and easy access to the brain's master brake pedal?